Hello, my dear students. Welcome you all to Sri Jayendra Rank Zone classes. My dear students, I am your mentor and your friend for the subject biology, and I will be taking the zoology section, that is animal science. My dear students, in the last lectures, we have completed the connective tissue, the muscular tissue, and the epithelial tissue, and now it is turn for the nervous tissue. Nervous tissue is basically the most evolved and most organized tissue in case of vertebrates, especially the mammals. So moving to nervous tissue. The origin of nervous tissue is from the ectoderm of embryo. So embryonal ectoderm give rise to nervous tissue. Understood? What are the different properties or the spatial properties of nervous tissue? The spatial properties of the cells of nervous tissue is excitability and conductivity. Mean excitability and conductivity, these two properties, they make this tissue highly evolved and organized tissue of vertebrates, animals. Although this tissue is also present in dormitory form in case of invertebrates, but in case of vertebrates, because of the development of vertebral column, the tissue chain takes a unique shape and a unique function. So, excitability and conductivity, these are the two main characteristics. This tissue basically helps to provide a state of consciousness. It helps to respond to different stimuli. The cells of nervous tissue, known as neurons, they are specialized for receiving stimuli and transmitting message. This tissue forms nervous system of the body and including the following parts. The nervous tissue is basically of four types: neuron, neuroglia, ependymal cells, and neurosecretory cells. I mean, nervous tissue is having four types of cells. First is neuron, second is neuroglia, third is ependymal cells, and the fourth one is the neurosecretory cells. These are the four types of cells which make the nervous tissue composition. The four types of cells of nervous tissue: neurons, neuroglia, neurosecretory cells, and ependymal cells. Neurons. The neurons, they are considered as basic structural and functional unit of nervous system. So neurons, they are considered as basic structural and functional unit of nervous system. A neuron is a nerve cell with all its branches. Neuron is formed from neuroblast. Neuroblast is the precursor of neuron. Understood? It is a structural and functional unit of nervous system. Neuron is the longest cell of the body. The neuron is the longest cell of the body. Structure. Neuron is basically consisting of two parts. One is cyton and one is the nerve process, which is known as what? Axon. So one is cyton, which is also known as cell body, soma, or perikaryon. And the nerve process, they are known as what? Axon or axon terminal. Understood? So the main structure of neuron is that it consists of two parts. One is cyton and the other one is the nerve process, also known as these nerve process, they also contain axon terminal, and nerve processes, they also includes dendrites. They also includes dendrites. So two parts of nerves, cyton and the neuron processes or the nerve processes, which include dendron, axon. Okay. Now, what is cyton? Cyton. It is also called perikaryon or soma or cell body. It is having a granular cytoplasm which is called neuroplasm. So it consists of granular cytoplasm which is also known as neuroplasm. Understood? Cyton, it is a large spherical and centrally placed nucleus. It is having a large spherical and centrally placed nucleus. It is a large it is having a large spherical and centrally placed nucleus oblique nucleolus. Third point, it also have many thread-like fibers known as neurofibrils, sorry, for the conduction. It helps in conduction. Okay. It also consists of number of small basophilic granules known as Nissel's granules. It consists of basophilic granules also known as Nissel's granules. Understood? These Nissel granules, they are formed from rough endoplasmic reticulum with ribosomes and are the sites of protein synthesis. Rough endoplasmic reticulum, rough RER, okay, and they help in protein synthesis. Understood? The neuroplasm, which is present inside the cyton or the cell body, it is having large number of mitochondria to provide high energy for impulse conduction. So, the cyton or the neuroplasm have large number of mitochondria to provide ATP for energy. Okay. This energy is utilized in conduction of impulse or the stimuli message. Neuroplasm may have melanophores with melanin pigment and lipochromes with orange or yellow pigment. They may have melanophores with melanin pigment and lipochromes having a orange or yellow pigment. Yellow pigment. A mature neuron has no centriole, so it cannot divide. Centriole is absent. Okay. The neuroplasm or the cyton, it is having a bar body, okay, which is often seen abutting against the inner surface of the nuclear membrane of cytons in females. So females, they have bar body. In cyton. This has been provided to transformed X chromosome, helps in the transmission of X chromosome. There are some neurons which are having different shapes, like flask-shaped, okay, and they are known as Purkinje cells. Flask-shaped cytons are known as Purkinje cells. Okay, these Purkinje cells, they are found in cerebellum of the brain. They are found in the cerebellum part. Cerebellum. Understood? So these are the different parts or the anatomical features of cyton. It is having a granular cytoplasm, which is known as what? Neuroplasm. It is having a large spherical and centrally placed nucleolus. It have many thread-like fibers, which are known as what? The neurofibrils, which helps in the conduction. It is having small basophilic granules, which are known as what? Nissl granules, which helps in the protein synthesis. Neurons have large number of mitochondria, which helps to provide energy in the form of ATP to help in the impulse conduction. The neuroplasm may consist of some melanophores and lipochromes. Melanophores having melanin pigment and lipochromes have orange or yellow pigment. Centriole is absent. Females, they have a extra X chromosome, which is known as what? Bar body in the cyton. Some cytons, they are flask-shaped, especially found in cerebellum, means brain. And these flask-shaped cells are known as what? Purkinje cells. Now, there is a diagram which is given to discuss the transverse section of a neuron. This transverse section of neuron says what? That the outermost layer is the connective tissue sheath. Just below the connective tissue sheath, epineurium is present. Then the nerve fibers, they are surrounded by a covering which is known as what? Endoneurium. And the bundles of nerve fibers, they are covered by a sheath which is known as what? Perineurium. As we have studied in muscles, epimysium, perimysium and endomysium. They have blood vessels. Okay. Now, it is having two parts. One is the axon and the another one is a cyton. Axons and dendrites, they come under the neuron processes. Okay. So you can see these are the axons. Then it is covered with a medullary fatty sheath. Okay. Medullary fatty sheath and neurulema. Okay. It has already been discussed. Yeah, all the points I discussed. Made up of messenger mRNA, endoplasmic reticulum, ribosomes, and has affinity for basic stain. That is why they are known as what? Basophilic granules. Understood? They are made up of mRNA, endoplasmic reticulum, and ribosomes. A mature neuron cannot divide because of the absence of centriole. Okay? Now, the nerve process, it is called neurite, which extends or which varies in the length. They basically extend from cyton, and they are of two types, dendrites and axons. Dendrites, also known as dendrons, and axon or axis cylinder, also known as neuroaxon. So dendrites and axon, also known as axis cylinder. Okay. Dendrites, also known as dendrons. Dendrites may be one or several. It is branched. Dendrites, they are the short tapering branch process. Short tapering branched process. Okay. The dendrites contain neurofibrils. They contain neurofibrils. Neurofibrils, which helps in the conduction. Neurotubules are also there. Neurotubules. They also contain nacelle granules and mitochondria. Nacelle granules are the basophilic granules and mitochondria. Mitochondria. Understood? They help in the conduction of nerve impulse towards the cell body. Then axon. Axon is a long single cylindrical process with a uniform diameter whose main function is to conduct the nerve impulses away from the cyton. So axon is of efferent in nature. And dendrites, they are known as what? Afferent in nature. Understood? The axon arises from a conical projection which is known as what? Axon hillock. So it arises from a conical projection. Known as what? Axon hillock. Understood? Axons, they are the longest nerve processes of a neuron. Giant squid, Loligo, has axon about 1500 micrometers in diameter. Neurons with very long axon is called Golgi type 1. Neurons of very large axon are known as what? Golgi type 1. Axon possesses only neurofibrils, nacelle granules, Golgi body, ribosome, fatty globules, they are absent in case of axons. So nacelle granules, they are present in dendrons but absent in axons. Golgi body, they are absent in axons. Ribosomes, they are absent in axons, and flat globules, they are absent in axons. Axon is usually branched only terminally into cylinder branches called telodendria or telodendrites. So axon, it is branched terminally to form telodendrites. Telodendrites or telodendria. Understood? This telodendria is having a knob-like structure which is known as what? End bulbs or axon terminals. The distal end is knob-like, known as axon terminals, or buttons, synaptic buttons, or synaptic knobs, or end plate. Understood? The synaptic knobs, they contain mitochondria and secretory vesicles. The plasma lemma of axon is called exolemma, where cytoplasm is known as what? Exoplasm. The axon ends in a group of branch terminal arborization. Ends of terminal arborization possesses knob-like structure known as synaptic knob, synaptic buttons, or end plate or axon terminal. The branch or the branches is known as what? Telodendria. Understood? The synaptic knobs consist of mitochondria and secretory vesicles. Synaptic knob consists of mitochondria and secretory vesicles. Understood? The synaptic knobs comes to lie very close to the dendrons of the next neuron to form a junction which is known as what? Synapse. There is a microscopic gap of around 200 angstrom which is known as what? Synaptic cleft. The synaptic cleft is basically a very small gap around 200 angstrom which is known as what? Synaptic cleft. Synapse is a junction of two adjacent or consecutive neurons. The synaptic knob, it come to lie very close to the dendrons and make a synapse. Understood? The nerve impulses are transmitted from axon to dendron with the help of a chemical known as neurotransmitter, which is either acetylcholine in case of skeletal nervous system, and adrenaline in case of autonomic nervous system. Understood? Acetylcholine or adrenaline is produced by the secretory vesicles of synaptic knobs. So acetylcholine is the major neurotransmitter in case of skeletal nervous system, also found in parasympathetic nervous system, and adrenaline, epinephrine and norepinephrine, they are the primary neurotransmitters of autonomic nervous system. Parasympathetic nervous system. Okay. Understood? Types of neurons. So types of neurons on the basis of number of nerve fibers: pseudopolar, unipolar, bipolar, and multipolar. Pseudopolar, which is not having any process, no nerve process. Unipolar, one dendrite and one axon, mean one processes. Bipolar, two processes, and multipolar, many processes. Understood? Unipolar neurons, when only one nerve process arises from the cyton, is a unipolar neuron. In case of unipolar neuron, it is a cyton, okay, which give rise to the exact nerve process, which only act as axon also, but there is no dendron. Okay, there is no dendron. So in case of unipolar neuron, axon is there, but dendron is absent. Absent. So in case of unipolar neuron, dendron is absent while the axon is there. They are found in early embryos. Unipolar, they are found in early embryos. Understood? These Unipolar neurons, they help in the transmission of nerve impulse, that too in a single direction, in one direction. Bipolar neurons, when there are two processes, these bipolar neurons and the two processes, one is axon and the another one is dendron. They are found in olfactory epithelium. They are found in olfactory epithelium. Okay. They may be isopolar or heteropolar, means the dendrons having irregular branches. So on the basis of that, it is given the name isopolar or heteropolar. Ganglia of 8th cranial nerve. So ganglia of 8th cranial nerve is having a bipolar neuron. Ganglia of 8th cranial nerve. Eighth cranial nerve. Okay, it is having a bipolar neuron. Understood? On the basis of number of dendrons and axons, neurons are of three types. It is already mentioned: unipolar, bipolar. Bipolar is present in the retina of eye, olfactory epithelium, organ of Corti, and taste buds. Understood? Multipolar neuron, when there are many dendrons and one axon. Axon is only one. Please remember, axon is only one. Dendrons can be many. And it is termed as multipolar neuron. Multipolar neuron found in the nervous system of adults. On the basis of functions, it is of three types. So on the basis of function, it is of three types. Sensory or afferent neurons, they connect sensory organs with central nervous system and brings sensory impulses to it. Motor or efferent neuron, they connect central nervous system with effectors, effector muscles, muscles and glands, and carry out motor impulses to them. So sensory or afferent, motor or efferent. Understood? Then there are interneurons which help to connect the sensory and the motor neurons, also known as adjuster neurons. They are present in the central nervous system, brain and spinal cord, and they connect two or more neurons for distance transmission of impulses. So on the basis of function, it is of three types. One is the sensory or afferent, second motor or efferent, and the third interneuron or adjuster neuron. Is it clear? Now before discussing about the nerve fiber, I would like you to, this is the picture in which the different parts are shown starting from the dendrites. You can see the branchings. These branchings, they are known as what? Dendrons. They are known as what? Dendrons. Now they have missile granules. This is the side neuroplasm or the soma cell body, which is having a nucleus as well as the nucleolus. They have neurofibrils and neurotubules for the impulse conduction. The part where action potential starts, it is known as what? Axon hillock. It is also having numerous mitochondria, means the neuroplasm which is present in the cell body. And this is the nerve process axon. This is the nerve process dendron. So it is covered with a sheath which is known as what? Myelin sheath in case of myelinated nerve fibers. The myelin sheath of Schwann cells. So Schwann cells, they basically form myelin sheath. There are some places where myelin sheath is absent and which proves to be conducting. That is known as what? Node of Renvie. Node of Renvie. Understood? Then the covering of Schwann cell is known as what? Neurolemma. In case of anastomosis, when nerve makes branching, it makes collateral axons, okay, especially in case of critical thinking. This process of making branches is known as what? Anastomosis. Collateral branches. Then this is a neurofibril nucleus of Schwann cell, myelin sheath, which is the fatty layer, fatty tissue layer, cytoplasm of Schwann cell, neurolemma of Schwann cell. So Schwann cells, they are basically framed or guarded by neurolemma. These are neurofibril nodes known as node of Renvie. The cytoplasm known as what? Exoplasm, and the membrane is known as what? Exolemma. This is the cytoplasm of Schwann cell, axon terminal or terminal arborization, or synaptic knobs or synaptic buttons. Understood? This is a typical nerve fiber. In medullated nerve fiber having myelin sheath, A is the medullated nerve fiber having myelin sheath, and non-medullated nerve fiber is not having myelin sheath, no myelin sheath, or not having myelin sheath. Understood? You can see the Schwann cells with the nucleus and the cytoplasm of Schwann cell. They guard or they cover the axon. This is exoplasm, and this is known as what? Exolemma. These Schwann cells, they cover the axon. Schwann cell surrounding part of axon. The part which is not having Schwann cell is known as what? Node of Renvie. The segment of myelin sheath formed by Schwann cell. But these are the different stages in the formation of myelin sheath by Schwann cells. Axon. Axon of a neuron is covered with one or two sheaths. On the basis of presence or absence of sheath on nerve fibers are of two types: medullated or myelinated, and non-medullary nerve fibers. So you can see this is the medullated nerve fiber having a covering, and where it is left, it is node of Renvie. The action potential in case of myelinated nerve, it jumps from one node of Renvie to the another node of Renvie. That is known as what? Saltatory conduction. Okay, it is known as what? Saltatory conduction. It helps in saltatory conduction. But in case of non-myelinated nerves, there is no myelin sheath. Okay, this is unmyelinated axon. Is it clear? The action potential begins at axon hillock. It moves in this direction, unidirectional. The current speed is the electrical event that triggers V-gated channels, and thus the action potential a tiny bit down the axon. Electrical current speeds, it maintains the flow of charge ions to open the channels, and these channels are ion gated. Understood? Now medulated or myelinated nerve fibers. In these nerve fibers, around a nerve sheath, a fatty substance is formed which is called medullary sheath or myelin sheath. Understood? The medullary sheath is not continuous, and the point of absence of medullary sheath is known as what? Node of Renvir. Node of Renvir. The part of the medullated nerve fiber between two adjacent nodes is called internode. The part of the medullated nerve fiber between two adjacent node of Renvir is known as what? Internode. The medullary sheath and the node of Renvir is surrounded by transparent cellular outer covering known as neurolemma of Schwann cell. The Schwann cells, they are basically covered by a membrane which is known as neurolemma, and or we can say the Schwann cell, they basically cover the whole of the axon membrane in case of myelinated or medulated nerve fibers, and that continuous covering is known as what? Neurolemma. Just beneath the neurolemma, a thin layer of cytoplasm lies which contains nuclei to form Schwann cells or the sheath cells at intervals. These nuclei are termed as nuclei of Schwann cells. So the nuclei of Schwann cells, they help to form the sheath or the Schwann, means which helps in the formation of or secretion of proteins or the fatty substances, which helps in the making a cover around the axon. Medulated nerve fibers, they are found in brain and spinal cord, cranial nerves and spinal nerves. So myelinated or medulated nerve fibers, they are found in brain and spinal cord, cranial nerves and spinal nerves. Understood? In the central nervous system, medulated nerve fibers form white matter. In central nervous system, the medulated nerve fibers, they form white matter. And the gray matter is composed of cytons. Understood? Now coming to next, non-myelinated or non-medulated nerve fibers. In these nerve fibers, there is no medullary sheath or myelin sheath. The axon is surrounded by neurolemma only, and just below the neurolemma, a layer of cytoplasm containing nuclei at intervals is present. The node of Renvir and internodes are not present in case of non-medulated or non-myelinated nerve fibers. The non-medulated nerve fibers exist in autonomic nervous system. Okay. Medulated nerve fibers, they exist in brain, spinal cord, cranial nerves and spinal nerves. In central nervous system, non-medulated nerve fiber, they are found in gray matter. Function, the nerve fibers are of two types, afferent or sensory, efferent or motor. Afferent fiber carries the sensory impulse from the receptor organs to the central nervous system, brain and spinal cord. So they basically helps to provide information to brain and spinal cord, and that is why they are known as what? Efferent nerve fibers. Efferent or motor nerve fibers, efferent fiber carries impulses from the central nervous system to various effector organs like muscles and glands. Is it clear? Now before talking about the neuroglial cells, I would like to write here, that's why I have added the page, the medulated and non-medulated nerve fibers. Difference. Medulated nerve fibers or myelinated nerve fibers, and non-medulated or non-myelinated. Non-medulated. The medullated nerve fibers, they are found in the white matter, brain, spinal cord, cranial nerves and spinal nerves. Non-medullated nerve fibers, they are found in gray matter of brain and in the autonomic nervous system. Okay, so they are found in white matter of brain and spinal cord. They are found in gray matter of brain and spinal cord. Non-medullated nerve fibers, they are also found in autonomic nervous system. And they are also found in cranial and spinal nerves. The medullated nerve fibers, they are covered by inner medullary sheath and outer neurilemma. Covered by inner medullary sheath and outer neurilemma. And the non-medullated nerve fibers, they are only covered by neurilemma, medullary sheath is absent. Only covered by neurilemma. The node of Renvir and medullated nerve fibers are present. Here absent. Node of Renvir absent. The diameter of medullary nerve fibers is more in comparison to the non-medullated nerve fibers. The color of medullated nerve fibers is white because of myelin sheath, and non-medullated nerve fibers is gray. Speed of conduction, medullated nerve fibers saltatory conduction, fast, and non-medullated nerve fibers slow. Collateral branches in case of medullated nerve fibers is present, and in case of non-medullated nerve fibers absent. Collateral branches absent. Understood? We're coming to next. Neuroglia or glial cells. Neuroglia or glial cells, they consist of supporting and packaging cells found in brain, spinal cord, and ganglia. Supporting and packaging cells. Brain, sorry. They are found in brain, spinal cord, and ganglia. They are non-nervous cells which lie between the neurons of CNS, ganglia, and retina of eye. So they are supporting and packaging cells. They are non-nervous, means they are non-conducting. They lie between the neurons of CNS, ganglia, and retina of eye. These are many times 10 times approx more numerous than the normal neurons. They have a fibrous astrocyte, a star-shaped cell, protoplasmic astrocyte, oligodendrocyte, and microglial cells. So the neuroglial cells, they are of mainly three types: astrocytes, fibrous and protoplasmic, oligodendrocytes, and microglial cells. Neuroglial cells, they are of following types: microglial cells, they are small sized cells which are spindle shaped. The microglial cells act as a defensive phagocytes. Understood? In central nervous system, they arise from monocytes. They arise from monocytes. They arise from monocytes. Astrocytes, they are highly branched and star-shaped cells which form blood-brain barrier, therefore only located in brain. Blood-brain barrier. That's why only found in brain. Only found in brain. Okay. So astrocytes, they are highly branched and star-shaped cells. They help to form the blood-brain barrier, and that is why they are found only in brain. The blood-brain barrier helps to avoid unnecessary entry of different byproducts or metabolically active or inactive products. Okay, so it basically act as a watchman or a guard to prevent the entry of toxic neurotoxic substances inside brain. Then comes oligodendrocytes. Oligo means small or few. They have few branched processes which resemble dendrons of a neuron. They form myelin sheath around CNS, axon, example Schwann cells. The Schwann cells, they are the example of, or they are the type of oligodendrocytes because they helps to form myelin sheath. Okay. Types of neuroglial cells: astrocytes, protoplasmic astrocytes. Different kinds of neuroglial cells include, this is oligodendrocyte, and this is the last microglial cells. Understood? The first is A, astrocyte, fibrous astrocyte, then the protoplasmic astrocyte, then the oligodendrocyte, and the fourth one is microglial cells. All the characteristics of all these four cells been discussed. Now coming to next, my dear students, function. Neuroglial cells, they help in packaging, they provide nutrition, they act as a phagocyte, okay, as in case of microglial cells, and consume microorganisms. Microglial cells, they are produced from monocytes. They help in memory process. These insulate the adjoining neurons. Then comes the next category, that is ependymal cells. They are cuboidal and ciliated. Okay. Cuboidal and ciliated epithelial cells. They makes a epithelial lining which is called ependyma that lines the ventricles of brain and the central canal of spinal cord. These cells are generally ciliated. So they are ciliated cuboidal cells which generally makes the lining of ventricles of brain, that is ependyma, and the central canal of spinal cord. Ependymal cells, they help in the circulation of CSF, cerebrospinal fluid. Cerebrospinal. Then comes the next category, neurosecretory cells. These are special type of neurons of hypothalamus of brain. They are endocrine in function and release neurohormones, which are releasing factors. Through portal system, hypothalamus, pituitary stalk system, to the anterior most lobe of the pituitary gland, which then regulates secretion of hormones like TSH, thyroid stimulating hormone, growth hormone, then LH, luteinizing hormone, ACTH, adrenocorticotropic hormone, follicle stimulating hormone, and prolactin. The neurosecretory cells, they are the special type of neurons of hypothalamus of brain. These cells are found in hormonal in function, they are endocrinal in function, and they release neurohormones. The pituitary and the hypothalamus is connected with the pituitary and hypothalamus. They basically connected with the pituitary hypothalamic stalk system, which includes the neurosecretory cells. These neurosecretory cells help hypothalamus to control pituitary and pituitary to control other glands. Okay. So now we are going to, I think this part is completed. Now we are going to talk about something which is to be added to your knowledge bank. So I will add one slide. Yeah. The nerve fibers the axon or the dendrons of nerve cell, they are covered with one or two sheath, which is known as what? Nerve fibers. Okay. So axon and dendron, they are covered by nerve fiber. There are two types already discussed, the modulated or non-modulated. Non-modulated, they are basically the non-myelinated, and modulated, they are myelinated. Okay. Now something I just want to discuss about the neurolemma. Please rectify the spelling. The neurolemma, basically it is a tubular sheath cells, sheath cells also known as Schwann cells. Sheath cells also known as Schwann cells. And these cells are placed end to end. The neurolemma is continuous over the node of Renvir. It is continuous over the node of Renvir. The function of Schwann cells is to produce myelin sheath. Around the neuroaxis. The medullated nerve fibers, they are present in brain, spinal cord. They lack neurolemma. So please remember, the medullated nerve fibers which are present in the brain and spinal cord lacks neurolemma. Understood? They have an incomplete covering of neuroglial cells. So the brain and spinal cord medullated nerve fibers, they are covered by neuroglial cells. Which probably produce myelin sheath. Neurolemma present around the peripheral nerve fibers enables them to regenerate after injury. Enables the peripheral nerve fibers to regenerate after trauma or injury. Like neurotomesis, okay? The nerve fibers in the brain and spinal cord could not regenerate after injury due to lack of neurolemma, and that is why the nerve fibers of brain and spinal cord cannot regenerate as they lack neurolemma. Understood? The medullated nerve fibers occur in the white matter nerve fibers, white matter of brain and spinal cord. Along with cranial and spinal nerves. So this is all about the nervous tissue. Okay. We have discussed everything except the physiology, means the function and the types of different nervous systems. So we will be discussing the nervous system and their types when we will study the human physiology. So please go through this slide and the previous slides. We have completed the chapter animal tissues. Okay. In the next turn, we will be doing the MCQ questions. Till then, my dear students, bye bye. Take care, everyone. Stay safe. Stay happy.